Real quick. Okay, there you go. Good afternoon and welcome to our recording here for Module 6 to see what to do in terms of the labs and our report here. So Module 6 is going to take a look at cellular division, the patterns of heredity. So you're going to kind of bounce off with what you've been learning in the lecture as well as here at the lab. You're going to look at the knowledge of or understand your knowledge of cells, cell division, heredity, how things pass through the generations, traits as well too. So looking at what favorable traits might be passed along through the generations here. So as you begin the module, just take a look at some of the reading material. The readings and resources is where you will get this information, which is your module lab. So first, let's go through what is due for this week, and then we'll talk in detail about the lab for this week here. So remember, I posted or I had a quick little warning last week, as well as I posted a message yesterday. Start your lab as soon as possible. If you don't start it past today, you are going to be late on it. So make sure you start it. The lab does involve seed germination, so it takes about 7 days. So again, you need to complete everything within the time frame. So here's kind of what to expect in times of preparation: 15 minutes of prep, 30 minutes for days 1 and 4. About an hour or so for day 5, and then 30 minutes for day 8. There's also a second lab to do as well too. So there are no like lab that goes along with this particular one, the monohybrid and dihybrid cross. You're going to go to this website called the Biology Project, and you're going to complete the entire 13-question problem sets for both monohybrid as well as dihybrid. Make sure that you are answering the questions from the lab report based on what is in that website here. It's recommended to use the days that you're not collecting data, so those in-between days, to work on this lab. And again, make sure you use the lab report. Okay, so again, just two labs that are due for this week. So with that, let's take a look at the Mendelian Genetics lab. So this is the lab that you'll be using. I took everything out already, that way I can show you. But again, Mendelian Genetics right here, lab 6. So the material that you'll be using, again, there's a real quick intro about genotype, phenotype, what's dominant, what's recessive. So take a look at that. Also about Punnett squares as well, and genotype. So again, take a look at that. The material, so you'll be using 3 seeds. They're pre-labeled for you. So you have P1, F1, and F2. So make sure you are using the correct seeds to go with it. Make sure you have the metric ruler that should be in your other lab with the graduated cylinder and everything. I don't have it offhand. Your forceps and beaker. You have Petri dishes. I didn't take them out of the bubble wrap, but they look like this. So you should have 1, 2, 3 here, and then 3 here. So make sure you have 6 of those. You should also have some filter paper as well as a graduated pipette. Okay, so this is all for lab 6 here. Go ahead and put them up before I accidentally break anything. Also needed but not included in the lab kit, so make sure you have some water, so tap water, 200 mils to use, a fluorescent light source, black light's fine, or some other type of lighting, pencil and scissors. Just note that these fast plant seeds, they are GMOs, genetically modified organisms. They're engineered to grow rather quickly, which is why, again, you should be starting the lab very soon here. So it is, if a common incandescent light source is used, the heat from the light may cook the seeds and they may not grow properly. So again, make sure you use some type of cool light source. A fluorescent light, LED light is totally fine as well too. I think most of us have LED light, so it shouldn't be too much of a hassle to find some type of cool light source to use. So don't use regular incandescent because it might overcook your seeds and they may not grow fast enough. Okay, so make sure, again, you read through everything first and then complete the lab. Don't forget, no eating, drinking, chewing gum or anything. Make sure you have a clean work area. And also these fast plant seeds are very, very tiny. So make sure you use the forceps to help position them. Okay, so day 1, here you're going to plant in this activity plants from one of the true breeding parent populations from the first generation will come up here. So you're going to be planting P1 and F1 generations. So it has some instructions here about cutting the filter paper, placing them on the filter paper, and then cutting it and putting it in the Petri dish. So make sure you follow the directions here about doing that. And then once you complete activity 1, it's just kind of a wait and see. So by day 4, take a look at your seeds, examine them for the purple stem trait and the dwarf stem height trait that we're going to be showing or exhibiting here. Here you're going to count, and it has here a good photo here to kind of gauge your plant growth and coloration and everything. So you can count the number of plants that have a purple stem and dwarf height, purple stem normal height, non-purple stem, which is green stem, but they're small, they're dwarf, or non-purple, so they're green and normal stem height. So you record your data table 1 and the percentages, and then you're also going to make a prediction. Day 5, you're going to plant the F2 generation. So remember, we did P1 and F1. Now we're going to do the F2 generation. Same thing, fill the bottom of a Petri dish with water. So you're going to cut strips again. Use a pencil to label out the uncut ones. So just follow the directions here. And then you can put them under the light source, that fluorescent light or that cool light source. Again, not incandescent lights. Day 8, you're going to do something very similar to what you did in day 4. You're going to record your information in the data table and then pretty much clean up. So again, here is the data table 1 that you're going to do for your first experiment, looking at the P1 and F1 generation, looking at plant height and stem color, either green or purple. And then F2, also looking at purple stem or non-purple stem and height as well, okay? So again, pretty short and simple lab. The only thing is to start it early because you're waiting for these things to sprout. So again, don't start this Sunday night at 10 o'clock at night because you're not going to get the results whatsoever anytime real soon. So make sure you are starting it. You should have already technically started it. So if you haven't started it, make sure you're on it, okay? So again, the only thing that are due, of course, is the Mendelian genetics, and then your second lab, monohybrid and dihybrid cross, looking at the website, and then filling out the lab report for that one. So just 2 lab reports, okay? Let me know if you have any questions, and I look forward to reviewing your lab reports and seeing the great results you guys get from this experiment this week.